Wait, let me
That
just look for a second.
Looks
I think-
good.
That looks perfect, Carolina.
Yeah.
Beautiful.
Setting up done.
What do you mean you're going live?
Hmm?
Streaming on YouTube.
Gotcha.
Is there a
If
little
I-
progress bar?
If I go, yeah, so it's done, but if I go to the link, I don't see anything.
Yeah, it takes, probably 20 seconds or so for it to start streaming.
Oh, really? Okay. Because I see live now the eOpen stream, but that's about it.
Yeah, that's what's always streaming. I see it now. Yep.
You see it? Okay.
Yeah, just
Yeah,
refresh.
I have one comment, which is that somebody told me that yesterday the speaker was blocking the slides on the live version or something, and what didn't happen the previous 2 days. I don't know what that's all about, but I just thought I'd tell you guys.
Oh, I think if they don't stop their video, they'll appear in a small window that overlays the corner of the screen. So you may want to stop your video while you're showing your slides. Yeah.
How do you want to do this? Should I stop sharing now and then start sharing after? Or
Sure,
just...
go ahead, and then you can
Alright.
start sharing after I introduce you. That sounds good.
Hey, Richard, I still don't see it. You see it?
Yep. Did you refresh your window?
Yeah.
Yeah, I'm definitely seeing it. Let me take a look real quick.
Let me see. Well, I'm going into incognito. Maybe there's some cookies cache type of thing. Nope. I mean, we haven't had more than 500 anyway, so like at least we didn't have yesterday. That's that many people. I mean, this is fantastic to have it as a.
Just give me 1 second.
I mean, if we don't have 1,000, I don't think I can speak.
So Richard, you need to go to videos and then to live now. Right? Like the link is different.
Oh, is it?
Yeah.
I see it.
Yeah.
Okay. I don't know. So the link, well, never mind, because that's the link that I'm actually.
If we don't have 1,000, I don't think I have to hear myself say that too.
Yeah, that's pretty embarrassing.
All right. Should we start letting people in the meantime?
No,
Or maybe wait another
I'd
minute.
wait another minute.
Safe.
Yeah, because otherwise they'll just sit there and hear us talk. Yeah, I guess you could now. I guess it's not. That's 8:59. That's fine.
Yeah, exactly. All right. Okay, I'm gonna start recording and let people in.
Right.
I would tell a joke,
Morning,
but
everybody.
I only know.
No, don't do it. Don't
kids
do it.
joke nowadays.
Don't do it.
We're live on YouTube, so.
Whatever you think it is, it's not. It's not as funny as you think it is. It could be dangerous.
All right, everyone, I'm going to go dark now.
Beautiful. Thanks,
All
Richard.
right. Thanks again. Appreciate it very much.
Hi, Julie.
Hey, Carolina, how are you doing?
I'm doing well. It's nice to see you.
Okay, we'll start in another minute, everybody, just so you know where we're headed. Still waiting for people to join. Good morning, everybody. We'll wait another 30 seconds or so, and then we'll get started. Thanks for tuning in. Physical Biology, the Cell Remote version 2020. What a bummer. so much more fun, Carolina, if we could go for a swim today.
I know, I was just gonna say it's, I mean, I guess we could both go swimming, but.
I already did, but it wasn't. It wasn't the same thing, shall we say.
Zoom swimming, you can swim with water resistant headphones and
That's right.
keep talking.
All right, I think we'll get started. So good morning, everybody, and thanks for tuning in, as I said a few minutes ago. So it's my great pleasure to introduce today's speaker for Physical Biology, the Cell, our course, Carolina Tropini from the University of British Columbia. She fits perfectly into the kinds of talks that we love to have in our course, and I think you'll see why as the talk unfolds. But let me just say a few words about her background. I'll start with the facts, and then I'll turn to some of the more interesting features. So first of all, I first myself met Carolina, I don't know, 15 plus years ago when she was an undergrad at the University of British Columbia, where she's now a professor. She was in the lab of Carl Hansen, and as it happens, Carl was a grad student in applied physics at Caltech, and I was on his committee. He was with Steve Quake, and so I went up there to visit him and had the pleasure of meeting him. After that, she went to Stanford for grad school, where she worked with Casey Huang. And so I should say that when she was an undergrad, she was working on microfluidics and digital PCR and that kind of stuff. And then with Casey, she worked on topics that merged mechanics and microbes in very interesting ways. Things like, what's the nature of growth in the presence of osmotic pressure? Or alternatively, how do bacteria grow in the presence of some sort of constraints? And then after that, she was a postdoc with Justin Sonnenberg, also at Stanford. And if you look at the publications that they've written, it's super interesting because I think it's a very important theme for biology writ large, which is, in a way, how does mechanics and physics meet the genome? How does the mechanics and physics meet ecology and development and so on? And so she was interested in the way that physical perturbations would alter the microbiome. I think it's really a glorious sort of thing where it merges what we know about things like metagenomics with things that we would think of in terms of physics. Her connection to MBL is quite long and important. She was a student in physiology, I think. I don't know. Again, I have no idea how long ago. And then after that, she also served as a TA with me, and we had a blast. Both of those summers that she was there, we swim all the time. You will not be able to guess it by listening to her, but her native tongue is actually Italian, and I have had the great pleasure more than once actually of translating things with her. So we translated my favorite all-time short story so that my dad could read it after his big surgery. And when he woke up, I had promised him that I was going to read him the short story in English, which did not exist before then, and that was a great time, and I'm so appreciative for that. Anyway, Carolina's done all sorts of interesting stuff. She's got a very interesting personal life, and I'm not going to comment on that, just in the sense of lots of interesting hobbies and thoughts and things like that. And at any rate, it's great to have you here, and looking forward to your talk. I will let you know at about 5 of 10. And along the way, if we have any really important questions that are clarifications or something, Aaron will probably chime in for those. And then for the audience, I just want to alert everybody that we probably won't have time to get to all the questions, but we will send Carolina a list of the questions afterwards, and you can always send her a mail or whatever as a follow-up. Anyway, thanks again for agreeing to join us, and looking forward to hearing your talk.
Thanks so much, Robin. I'm so excited to be here, even though it's remotely. That was my most favorite trip planned for the year, honestly. Rob talked a lot about swimming, and this was a lot of the inspiration that has come from this work has really come from some of the chats that we've had, and I really hope that in the future we'll be able to do something like this again in person. So what I really want to talk to you about today is this idea of how we can think about the gut microbiota as a physical system that is really embedded in its environment. Somehow this is something that has not really been talked about from the perspective of thinking about the microbiota. Generally, when you hear about the microbiota, you hear about how it's affected by diet, how it's affected by the interactions with the host. But all organisms live very tightly connected to the physical environment. This is something that when we think about it from a perspective of a macro scale, it's really obvious. We can't think of an ecosystem without its habitat. And if I were to tell you that I want to take a cactus and embed it in the tundra, you would right away have a hypothesis as to what would happen and what conditions you might need to change to affect the survival. We just don't have this type of understanding for the microbiota. If you think of some of the procedures that are done right now in terms of trying to understand these systems, for example, fecal microbiota transplants, these are really ways in which we can transplant an ecosystem into a different habitat, and oftentimes a habitat that is diseased. And we would like to have a similar type of predictability and a similar type of understanding for the bacteria and viruses that affect this world. So when we think about the physical ecosystem of the gut, there's a lot of different niches that are present. So here it's a depiction of the mouse gut. In the case of a human gut, the cecum is much smaller, but there's a similarly type of laid out of types of environments. The environments are set up largely by the host, so either the human or the mammalian, or any of these systems that have a gut will have this type of different environments. And the host controls things like pH and oxygen permeability. So of course, when you think about pH, you'll think about the stomach that has an acidic pH, but there's also a lot of other controls of pH that happen throughout the gut. There's some really beautiful symbiotic relations between some of the short-chain fatty acids that are produced by the bacteria that get absorbed in the distal colon and they get exchanged for bicarbonate, which increases the pH. There's a change in a gradient of oxygen tension that comes from having a very oxygenated early or in proximal part of the gut to having a completely anaerobic distal part of the gut. There's also very large changes in nutrient availability because there's easy nutrients that get taken up by the host in the early part of the gut. And then by the end of the gut, where there's the higher part of the bacterial load, there's only fiber left and this complex carbohydrates that we're not able to break down, but we take advantage of the machinery and processes that these organisms can take on that we're not able to do. They will give us more energy. So of course, because we have these different environments, also the type of organisms that are able to live here become very different. We have bacteria that are very good at breaking down the tough fiber in the distal part of the colon, while we have other types of bacteria that are, for example, better at interacting with the immune system that in the earlier part of the distal tract is going to be trying to battle for the easy nutrients. These are also bacteria that are very good at surviving at higher oxygen concentrations. Because there's these larger scale gradients, we also find that there's a lot of structure happening at the micro scale. So here I'm showing you an image, a micrograph of the distal part of the gut in a mouse. And what I would like to really show here is how these microscale environments become really important. So at the bottom, we have this host epithelium, which is the part of the gut lining. And this gut lining, so here it's highlighted in blue, it's a DAPI stain. It's also producing mucus. There's goblet cells that excrete this very mechanical structure that provides both a mechanical separation between the microbiota that here is highlighted in yellow and red with fish staining, so that the microbiota does not penetrate and invade the host epithelium, but it also provides a food source. So you have this dual relation because the host actually really wants to have the microbiota being happy and fed. And if you don't eat the fiber, which are these large structures in red, the microbiota is left consuming the mucus. This is the symbiotic relation is really important, and it's really tightly linked. For example, for mammals that undergo hibernation, the mucus layer is basically what sustains the microbes during the times of the animal is hibernating. So this interaction is very important, and we need the microbiota as much as they need us. And by setting up these different environments, we make sure that the type of functions that are important for our well-being and our survival are being keeping this microbiota alive. However, these environments are very different, and when we study the microbiota just by sampling the stool, we lose the spatial organization that becomes really important for the biology of these systems. And importantly, there's also another aspect of this in terms of how the physical environment changes. Importantly, because whenever there's disease, the physical environment is affected. So here in the lab, there's a lot of different aspects that we care about. We care about peristalsis and flow in a way in which it changes the accessibility of specific nutrients. But there's also other types of factors in the physical environment that you might be thinking about. For example, changes in temperature can happen at the systemic level if there's a fever, but they can also happen at a very localized level if there's inflammation or if there's a cancer. These changes in temperature have been known for a very long time to affect very strongly the way the bacteria can grow. Very early experiments in E. coli showed that even just a half a degree of temperature can affect the growth rate by tens of %. There's also changes in pH that can happen because of inflammation. In the case even of over-the-counter drugs that people take very commonly, for example, antacids. These changes in pH very strongly affect the way that bacteria are able to process different carbohydrates as well as interact with each other. And what I'm going to talk to you mostly about today is this idea of osmolality. So this is another force that is very important. Osmolality refers to the concentration of particles, but more than a concentration, it refers to the osmotic pressure that due to the number of particles that are present in a solution, organisms will experience. And osmolality is something that is very tightly controlled by in the serum, in the blood. And it's something that also very strongly impacts the way that compounds can be exchanged in the environment of the gut. Osmolality is very strongly changed in the case of any type of malabsorption. So in the case of celiac disease or the use of laxative, as well as in a lot of inflammatory bowel diseases. Why this is important that we study it is because we've done everything that we've ever been able to fix the environment, to ensure that whatever we're growing will actually be able to live and grow as quickly and as well as it could. So whenever we look at the conditions that we have in the media that we grow cells in, we fix the pH, we fix the osmolality, we fix the temperature because we put things in incubators, we fix the material stiffness, the oxygen tension, so that we really optimize the way that these organisms are growing. But this is not representative of what is actually happening in the physical system. And the reason why this matters, I think it's more obvious at the macro scale. When we think about global warming, we're talking about decades of lifetime in which there's been an actually relatively small change in temperature. We're only talking about a couple of degrees of difference. But with these changes, we're seeing that over 1 million species are facing extinction. So now if you think about the lifetime and the timescales that affect these microorganisms, they have a life cycle in the order of tens of minutes. So if you imagine that you have a fever, it's only a couple of degrees of temperature difference again, but it can last for days. And this type of chronic changes will really affect the way that organisms can survive. And we can see also very large extinction events that are happening because of these perturbations that in our eyes are very acute and very transient. So to give you a sense of as to what you might have to think about with this type of perturbations, I wanted to just go through a thought process. If, for example, we're thinking about a change in temperature. So let's say that there's a fever. If a patient or a person has a fever, what are the important scales in the important length and time scales that will be affecting this system? Of course, changes in temperature will affect the electrostatic interactions between molecules. So we really need to start thinking about these effects at a subcellular level, but we're going to start thinking about this from a more of a cellular perspective, even though there's going to be changes that happen at even much smaller scales. So subcellular processes are going to be affected. There's going to be a heat shock response, and we're going to be able to measure this, for example, by looking at gene expression changes. So this heat shock response will also affect the way that these bacteria are growing. And importantly, is also going to affect the way that other microorganisms will be interacting with our bacteria that we're interested in. So we see, for example, that in changes in the physical environment, they very strongly affect the way the bacteriophages, which are viruses that infect bacteria, that are going to be able to actually interact with them. So all of these changes will impact the way that the bacteria can grow. And so here, when we think about the way the bacteria can grow, we generally think about these optical density curves in which we can see, look at the bacteria as they're growing over time. And in the gut, what it's going to mean is that you're going to have individual bacteria that are going to be now growing at different rates, which then will change the community composition. Because the community composition is changed, you're also going to have different molecules being produced. And so now this level now starts interacting with the host. Because different molecules are being consumed, the host is not going to be able to be exposed to the same type of molecules that is exposed to during health. There's going to be immune responses, there might be inflammation. We also talked about this mucus layer. The mucus layer becomes very strongly affected depending on the motility of these compounds down the digestive tract. And now you can see that as we go up the scales, there's going to be larger and larger effects, which are also going to be feeding back onto the subcellular processes. So in the case of fever, sometimes there are changes in stool motility, in water content, dehydration, and then all the way up to the larger scale, we then will have differences in the way that the organisms and the hosts themselves are going to be able to interact together. And this will affect the way that they might be exposed to other bacteria. And so this whole ecosystem is being affected. The reason why I'm talking to you about this is to just kind of give you a sense as to when we talk about broad spectrum perturbations, this type of physical perturbations that are so common in our day-to-day life have a really large impact in these communities. And what I think that is very important is that in studying these complex ecosystems, the standard approach of enumeration by sequencing is just not enough. We really need to have a multi-scale approach that combines not only our understanding of who's there, so these omics that tell us what species are present or what are expressing, but we also need to actually be able to understand the ecosystem, doing imaging, doing genetics, so that we actually change the bacteria to see how they're going to be responding. And in taking more an approach that that allows us to do smaller perturbations so that we can look at how the system responds and actually use computation to understand the changes. So here now I'm going to start talking about osmotic changes. And if anyone has any questions up to this point, I actually really like being interrupted so that it's not like a monologue. And also at this point, I probably don't even know if there's anyone connected anymore. So if there's any questions, I'd really love to hear them.
Tell you that, yeah, there's 220 people here and we hear you fine. I don't have any questions at the moment. Maybe Arnon does.
No, I don't. I think this is great, and you know, there will be questions starting to pop up in the Q&A, and
Okay,
you know, we'll see. You know, we'll see whether we interrupt you or not, depending on whether it applies. You know, it helps with understanding right away or not.
All right.
I guess
great.
I guess you know. I guess one quick question is, you know, that I choose as comment on the transport of water across the epithelium due to changes in the microbiome.
Yeah, so that absolutely has a really big effect. And we'll talk a little bit about this with respect to the osmotic imbalances. So changes in water content in the gut and dehydration very strongly affect the microbiota, because then it has to start undergoing, finding ways in which to either pump the water out, or in the case of stronger dehydration, to actually be able to get water in. And so we'll cover some of this when we're thinking about these osmotic imbalances. So why osmotic imbalances are particularly important for bacteria is due to the fact that they have, bacteria tend to have very high internal turgor pressures. So if you think about E. coli has about an atmosphere of pressure, which is about the level of pressure that you might find in an inflated bike tire for a road bike. So it's quite a significant amount of pressure. And there's even bacteria that can go up to hundreds of atmospheres of pressure in different parts of the ocean. Why this matters is that when there's an osmolality that cannot be absorbed, the only way that the osmotic balance can be kept in track is by having water flow out of the cell. So this causes changes in morphology, but you also imagine that this causes changes in the way that molecules pack together. Now, why we started becoming interested in this was actually we were using osmotic perturbation as a way to measure the stresses that were present on the cell wall. So this was during my PhD. I was very interested in understanding how the organization of the cell wall material was affecting the way that shape was being made. And so by shocking the cells with, this was mostly the E. coli cells, with half a molar sodium chloride, I was able to measure the decrease in size and then get information about the strains and the stresses that were present in this system. So one of the things that was really amazing to me during these studies was the fact that half a molar sodium chloride is about the level of salt that you might find in the Dead Sea. This is in the order of tens of atmospheres of pressure change that it was happening to this bacteria. And these E. coli cells would undergo plasmolysis, so this process of detachment of the membrane from the cell wall, but then they would be able to grow again. And so now if you put this in the context of an entire community of bacteria, you've now completely changed the rules of the game. And in systems that are so highly competitive, it becomes really important what your ability to grow into these systems and how it's changed by these perturbations. Why this is also relevant is because not only are there changes in morphology, but there's changes in the growth rate, as I was saying. And these are changes that you can see very directly by looking at these cells in microfluidic devices. So this is a cell that now is highlighted in green, and we can do this with thousands of cells at a time, and look at what happens when we're flowing different types of media. So we can track different types of the morphology. And here what you can see is that when we monitor the length, we see that the cells increase and grow exponentially. And as soon as the external osmolality hits the media, then the cells shrink. They shrink, but then they're also able to grow again, and they grow with a different growth rate. So we were really interested in understanding in how the short time scale growth would affect the long time scale impact on the entire community. And here again, now to go back to highlighting some of these mechanical features, one of the things that is really beautiful about these cells is that they're completely elastic. So you can go through the switches of having the osmolality on and off and repeat this for tens of times, and the cells are always able to regain their shape. And importantly, for these systems, these are some of perturbations that these cells have to go through during colonization, as well as during times of feeding different molecules. And as we'll go through even more so now that we have a world that is very highly using a lot of over-the-counter drugs. So in terms of osmotic perturbations, one of the most common drugs that is used in the US is Miralax. So this is the top 2 digestive remedy in the US. And the fact that 5.3 million kilograms are sold per year should be at least, well, I think it's quite surprising given that the standard dose is only 17 grams. So this is a really type of perturbation that the microbiota, or our microbiota as a community, sees a lot of. To study this in a system that we can monitor across the scales, we turn to these mouse models of the microbiota. And these mouse models have this really exciting capability that we're able to add to them very specific microbes. So these mice are derived to be germ-free by a C-section, so they don't have their own microbiota, and we can then expose them, inoculate them with specific bacteria to really try to get at the mechanism as to how different species interact with the host, as well as with different types of perturbations. We can also look at complex communities. So this can be communities that, for example, come from patients, or that come from different donors, as well as animal donors. So here in this type of setup, to try to understand the impact of osmotic perturbation, what we did was to use Miralax, which I'm going to be referring to as PEG for the rest of the presentation. And we exposed these mice to 6 days of this polyethylene glycol, which is this non-absorbable solute that draws water out from the epithelium and causes this increased flow. So this is the way that, in the same way that we saw that with the bacteria, water flows out of the cells, Miralax pulls water out of the epithelial cells and causes this increase in motility. And we then looked after this period of exposure to PEG, we wanted to see what the recovery looked like in these systems. One of the first things that we wanted to do, and I would like to say that this is a gut check, but this joke doesn't work very well because no one's laughing. However, we thought that it was something important to check was whether the osmolality actually was impacted. And what you might think is that the system is going to be balancing this perturbation because it's drawing water out of the epithelium. And we were interested in finding that actually, even though there's this increasing motility, this change in osmolality is not actually matched. So you can see here how the blood osmolality is kept at a very tight level, despite these mice becoming dehydrated over time. And this is something very, very important for mammalian life. Even by changing this osmolality level by a couple of percent, you would cause the death of the animal. And most animals have this very, very, very flat level of osmolality that most of the compounds that we use in the lab are actually matched to this. So for example, PBS has an osmolality that is exactly matched to that of blood. And again, here when we also look at the range of the healthy gut osmolality, this is in the range of all of the media that we've tested. So these are properties that we never purposefully tried to match, but as we created different media that allowed the cells to grow better, we actually ended up going into the realm of the healthy gut. And what we see here is that during the PEG treatment, the osmolality is almost doubled. And it's really going to be changing this physical environment that the bacteria and the microbiota generally is exposed to. One of the things that people always think about in terms of colonic lavages and use of laxatives is that they're going to do a cleansing. And this is not at all what we found. When we look at the colony forming units, so this is by plating the microbiota, we find that not only the density, but also the overall content of the microbes that we can culture is not decreased. In fact, there's a light increase. And at this point, what we wanted to understand was whether the composition of these microbes was changed. Of course, when you do this type of perturbations, what you expect is that the microbes will be responding. And there were two types of behaviors that we were interested in characterizing. One of them were the transient behaviors. And this was a lot of the different species of the microbiota. We can survey hundreds of different species. We found that there was a large proportion that underwent these transient behaviors, and some of them were not unexpected. So gamma proteobacteria, these are your typical E. coli, Proteus. They're bacteria that are very good at responding to stresses. And in fact, they became, when we look at their relative abundance, they increased in their levels. We also found this interesting negative correlation in the prevalence of specific bacteria at different levels of the phylogenetic tree. So if you're in the same branch of the phylogenetic tree, if your closest the genetic relative increases, you have a higher probability of being decreasing. So that we think that this is speaks to the availability of specific niches that then get occupied by taxa that are more similar. Importantly, we also found that there were some permanent changes. So here we're looking at a humanized community. So humanized mice are mice that are inoculated and only have a human microbiota. So here there's going to be in this talk probably about 20 people that are listening to this have S24/7, and every one of you has Bacteroidecia. These are a very common bacteria in our microbiota. And you can see that in this community, they make up a very large fraction. So S24/7 starts out at about 50% of the community, and the Bacteroidecia start about at 20%. When we apply the polyethylene glycol, the spec perturbation, S24/7 becomes undetectable. And here we see again a Bacteroidecia, which is within the same order as S24/7, that fills up this niche. Importantly, what happens is when we take away the perturbation, we've actually had a change in which we've depleted 50% of the microbiota. Why this is quite shocking to us, and this was actually not meant to be a pun with the osmotic shock, is the fact that not only are we modulating half of the microbiota, but we're changing an astounding number of bacteria that are present in this community. So this community have densities of 10 to the 11 bacteria per mil. This is a really incredible number. And not only are we modifying 50% of the microbiota, but we're taking away an entire family of bacteria. So to give you a sense of what a family of organism means is that if I told you that in an ecosystem, we were taking away all of felines. This is, from an ecosystem perspective, is a really large change. And these families have very specific characteristics. And we're now trying to understand the impact of S24/7 on the microbiota itself. But from the perspective of the community, this is a very, very large shift. So this, one of the questions that we also had was trying to understand whether this was something that was specific about S24/7 or whether it was something that was specific about the community. So we've since tested a number of communities that have S24/7. So here on the right, we have a community that's adapted to mice. It's a mouse microbiota. And we also see that S24/7 undergoes extinction in this sense. We can also do this even with just 2 members. We can drive S24/7 to disappearing even in the presence of very far fewer bacteria. So we think that this is something that is very specific about S24/7 itself. Now, the question really became to trying to understand the mechanisms as to why S24/7 would be driven to disappearance. And one of the obvious first places to look at was whether the osmotic perturbation was affecting its growth. And here we can take advantage of the fact that when we're thinking about physical perturbations, by nature, we need to find effects that are independent of the host being present. So we can do these experiments in vitro, and effects that are really dependent on the physical environment will be things that we should be able to also observe. Here, one of the things that we wanted to do was to also try to separate the effects from the osmotic perturbation to those of different types of osmolytes. So you can imagine that you will have the same type of osmotic perturbation if you were shocked with sucrose than if you were shocked with PEG. But in the long term, having the sucrose can actually improve your growth because of the availability of an extra sugar. So here we looked at 3 different types of osmolytes: sodium chloride, sorbitol, and PEG. And what we find is that when we look at the growth rate of S24/7, we find that it's very strongly impaired in vitro. And at the level that we normally see these bacteria growing in vivo with osmotic perturbation, they're not able to actually have any growth. So
Hey,
here,
Christina. Oh, sorry, Carolina. My question to you is, why osmolality instead of osmolarity? So somebody else asked that, and I actually was curious about the same
oh yeah,
thing.
that's an excellent question. So osmolarity can, I mean, for one, coming from a physics background, I think osmolality is a much more basic measurement. So if you think about a solution of 1 molar sodium chloride, because the sodium and the chloride dissociate, they actually have twice as much the osmolality as a 1 molar solution of sorbitol. And why this matters from the perspective of the mechanics of the system is that each molecule independently will exert an osmotic pressure. So when we think about the system, it really matters how they dissociate. Does that answer your question?
No, not necessarily. I guess I'm still a little bit confused. So
The other part is
actually, somebody typed a question about this, but
yeah.
I guess I'm
Maybe
just trying to think
say
about
also another thing. We actually can't
Yeah.
measure the osmolarity in a mixed medium, because there's going to be molecules that are broken apart. The composition is too mixed. So when we measure the osmolality, what we do is that we measure the freezing point depression. So that we look at if a solution was pure, it would freeze a specific temperature, but as there's contaminants in it, these changes in osmolality will affect the way that the freezing point changes. So when we're looking at gut content, we don't know what the osmolarity is, because we don't know how many salts, we don't know how many proteins, we don't know how many things are being dissolved. So from a practical perspective, we have to have this proxy of how many molecules are present in the system. Does that answer your question better?
Yeah, thanks. I'll think about it a little bit more, and maybe we can talk about it afterwards.
So one thing to note here also is that there's this very strong linear behavior that happens, and we're not surprised to see that there's this decrease in growth rate because of the fact that we've optimized these things. And what matters in these systems is really the slope of the changes in growth rate with osmolality. So here, as you can see, we now tested a large number of different bacteria, and all of these bacteria have a negative effect. And you can see that some with increasing osmolality. And you can see that PEG tends to have an even higher negative effect than what we would expect from the change in osmolality alone. And we think that this is due to the precipitation that the PEG causes and potentially some effects to the membrane. So oftentimes PEG is used to precipitate proteins or DNA in normal biological assays, and we think that it has this extra toxicity to the cells. Bacteroides theriatomicron, which is the middle panel, is one of the members of the Bacteroideaceae family that completely populated this niche that was opened up by S24/7 going away. And here you can see that it's also doing worse than normal. And this is one of those cases that I think is very similar to going camping with friends. You don't really need to be faster than the bear. You just need to be faster than I guess your slowest friend to survive. And this is something that it's very important in these competitive systems in which you really need to have this competitive advantage. And even if you're doing worse, it only matters that you're doing better than the other members of the community. We also see factors that are important that have to do with the cell wall. So gram-positives that have a thicker cell wall tend to do a lot better in this type of perturbations. And we also see this in vivo. So here there's two students in my lab that are following up on this project, trying to really understand even deeper the mechanisms for S24/7 resistance or lack of resilience to osmolality. And one of the things that we've since found is that there's specific genes that are missing in the S24/7 genome that affects its ability to survive this. So here was the data that we've taken with metagenomics to try to understand at a community level which type of genes were being affected by the perturbation. And here we were looking at osmotic genes, and particularly the osmotic stress genes are strongly overrepresented during times of the PEG treatment. So once we actually zoom into this picture, we actually found that there's a specific set of genes that are overrepresented. So these are the betaine uptake and biosynthesis genes. Betaine is an osmoprotectant that is present in plants. So that's where it comes from beets, at least the name, but it's present in a lot of different plants. And it gets imported by bacteria and even by plants themselves to keep water inside them. So what we found that we thought was very important is that this betaine was differentially represented in our different bacteria. And one of the things also that I wanted to highlight is that betaine has long been known to have a very strong effect in the way that bacteria can grow in minimal media. So these were experiments done in the late 90s with E. coli, in which it was shown that the growth rate in minimal medium could be made almost as high as the growth rate in rich medium by just adding betaine. And betaine is not actually used as a food source. It's only used as a way to protect the cells from this osmotic shock. We were really interested in finding that all the S24/7 isolates that we tested do not have this uptake gene. Where, for example, Bacteroides theriatomicron, which is the bacterium in the Bacteroideaceae family that overcame this perturbation, has this. So we're currently doing genetics to try to really see the importance of the system in this bacteria and as well as in the gut. So to wrap up this part of the presentation, when we think about these perturbations, and I think that this is also has an important impact on from the medical perspective, is the fact that the microbiota is not resilient to these perturbations. If we do these perturbations for a short enough time scale, so if we do it, for example, as a colonoscopy type of procedure, it seems like these organisms are able to recolonize. But as soon as we have time scales that occupy over 3 or 4 days, so that we actually don't, we have enough depletion of these cells, we find that we can lose them for the long term. And from the perspective of mechanism, we find that the osmolality itself is something that impairs the bacterial growth in vitro. And we find that there's the specific genes that are overrepresented as well as overexpressed during the osmotic perturbation. So this is something that the bacteria really respond to directly. So maybe here, I'll pause for a second to see if there's any questions about the osmotic effects.
Let's see. There might be some questions. So, 1 second. So, one of them was, so somehow these questions are getting all mixed, but where did it go? There was something about the PEG affecting negatively the three bacteria you were mentioning more than other sorts of insults. Yeah, PEG seems to have a higher negative effect on all three bacteria as compared to other osmolytes. Could you elaborate why that is the case?
Yeah, so that's an excellent point. As I was mentioning, we think that polyethylene glycol actually affects the membrane of these cells. So when we do these experiments in microfluidic devices, we can see that there's changes to the envelope of the cells. And as I was mentioning, PEG is often used to precipitate proteins or precipitate DNA. So we don't know the exact mechanism as to how it's affecting them more negatively, but it's an effect that it's beyond just osmotic perturbation.
Perfect. Yeah.
Right.
So I think we should keep going, and
Sounds good.
I think we should keep going for now.
Here now, we also wanted to understand non-osmotic effects. And again, this is going back to the idea that these perturbations go beyond the physical parameters that they're changing, and they really end up really broad spectrum. One of the things that we wanted to understand is how the organization of these cells was being affected. So here we looked at smaller communities in which we could label with fluorescence in situ hybridization specific cells, and we looked at whether the way that bacteria were assembling together and whether they were making microcolonies was being affected. So here what we can do is to look at individually now, because the cells are labeled, to look at how their spatial interactions are being changed. And the way we do this is by using probabilistic approaches, by looking at the pixels for individual cells, and because they're labeled differentially, to basically look at the radius of proximity to the closest different cell. So for example, here, when we look at the probability for the red cells to be next to the blue cells, we have a smaller radius than for the green cells. We can then plot these as a probability distribution, and probability distributions that are broader will indicate cells that form larger microcolonies as opposed to ones that have smaller radii. And when we look at specific cells, we can then look at that not all cells will be affected similarly. For example, here the names are not important, but the Lactobacillus tend to maintain as a pretty well mixed environment with the Bacteroides. But when we look at the Bacteroides themselves, they actually form larger microcolonies during the PEG treatment. So this is the curves in red that we see that they become more spread out. And here we're starting to investigate whether some of the genes that are involved in biofilm formation are being affected. This is a topic that is quite controversial in the field of the microbiota, because it's not really clear whether bacteria are forming biofilms. And one of the things that's quite interesting about the formation of biofilms from a mechanical perspective is the fact that biofilms would actually make it easier for the transport of, and for the depletion of specific bacteria. So this was work that was done in a number of labs in the zebrafish that looked at how the formation of these larger structures would cause their depletion. And we're really interested in understanding as to whether it could also provide some level of attachment. But another part that was important to us to observe was whether the mucus was being affected. So in imaging the systems, we can go back and image the mucus layer. We can do this both by using lectin stains as well as using antibodies. And we normally have this very thick mucus layer. And here you can see on the left-hand side that there's this striated mucus, and this is mucus that is being consumed by the bacteria. And this consumption is being balanced by the production of mucus by the host. What we found during the PEG treatment is that this mucus layer is largely depleted. I won't have time to tell you about this, but this gives rise to a large number of adaptive and innate immune responses. It causes attachment of bacteria directly to the epithelium. And it's really a time, even though the tissue itself is not very strongly inflamed, it's a time in which this entire system has to adapt to very close proximity between bacteria and the epithelium. Importantly, once this perturbation is done, the mucus layer recovers. It tends to recover to the same thickness, so we can quantify this by looking at the thick mucus part and seeing that it has this dynamics and that allow it to start recovering within 3 days after the perturbation. But it's not completely recovered to the same way. So we find that probably because of the types of bacteria that are changing, the striation is not always present in the same way. So importantly, this has a different orthogonal perturbation to the microbiota that is independent of the osmolality itself. It's dependent, of course, on the PEG treatment, but it doesn't have anything to do with the osmolality. It has to do with the fact that now the mucus layer is not available. And as I mentioned, this is an important food source for some of the bacteria. So here we looked at a specific family of bacteria that is known to specialize in mucus consumption, the Veromycobacteriaceae, and a specific member, Akkermansia, that is quite common in Western humans, it's a very important bacterium for the consumption of this mucus. And we find that the dynamics are very tightly correlated to the changes in mucus. We also wanted to understand whether bacteria that could choose between consuming mucus or other carbohydrates, whether they will be switching to consuming different things. And here we looked again in a small community in which one of our members, Bacteroides therialamicron, again, has the ability of consuming mucus as well as other types of carbohydrates. So here we looked at the expression levels for specific glycoside hydrolases, which are these enzymes that are involved in the breakdown of the mucus, and we found that they were strongly downregulated during the time of the PEG treatment and then went back to normal levels during the recovery. So here we have this orthogonal perturbation. We find that the mucus changes, it affects mucus consuming microbes, as well as the genes that are being expressed in the system. And here I wanted to wrap up in thinking about more again the system from an ecological perspective. So we talked about S24/7 that started out being every other member of the microbiota being in S24/7, and trying to understand whether this family of bacteria could actually be reintroduced in these systems. So here we can take advantage of the fact that these mice are coprophagic. So they eat stool, and we're then able to do these fecal microbiota transplant experiments quite easily. Here we have a community that has about 20% S24/7. And what we did is after several weeks of equilibration, we added a control microbiota to the cage. And we wanted to see whether S24/7 would have been able to repopulate and recolonize the system. My expectation was that because the community is so complex, there's still hundreds of pieces being present, and this niche had been filled, that S24/7 would have not been able to penetrate. But this turned out to not be the case. S24/7, even within the first time point that we were able to measure, it was already detectable. And we found that even after taking away the stool, it was able to stably colonize this community to at least within statistical levels to indistinguishable levels from before. And importantly, this gave rise to the fact that for these organisms, it's not necessarily a problem that they're very sensitive as long as they're able to disperse really well. And I think that here there's some important lessons in how we think about these systems and how we think about sanitation. There's a lot of these systems that if we worry, for example, the fact that there's about 2% of kids are exposed to these laxatives because of problems with constipation, we can worry about whether they're losing some important bacteria. But the hope is that if they're able to be reintroduced in their system, we may be able to actually compensate for some of these changes. And of course, right now we don't know yet whether S24/7 is good, bad, or just an average bacterium. But ecologically, we think that it's very important that we try to see where it fits into the picture of the interaction, at least with our health. So in general, one of the things that we became really curious about was where S24/7 was present in humans at a population level. So I mentioned that there's the bacteria that we have are quite diverse, but they're actually not quite as diverse as they are in traditional populations. So here what I'm showing you is it's a picoid plot that shows the similarity of microbial communities. So here, points that are closer together indicate communities that are more similar to each other, as opposed to ones that are further apart are communities that are more divergent. And on the y-axis, I'm showing you different types of populations that have this community. So these are human populations. We find that traditional populations have not only much higher diversity, but they also have bacteria that are quite different from the ones that are present in the industrialized populations. And we're now starting to understand how the impact of antibiotics, of different modes of birth, and exposure to low levels of fiber are really impacting the type of microbes that we have. And these effects are very strongly propagated because we're only inoculated by fewer bacteria because we're becoming very clean. And now in the light of this pandemic, it's also becoming very obvious that there's, of course, an important place for this cleanliness, but it's also affecting other parts of our health. So traditional populations never knew of inflammatory diseases. They don't know of allergies, and obesity is not a problem, while these are problems that we're finding a lot in industrialized populations. So one of the things that has become really obvious, for example, is kids that are more exposed to dirt and are more exposed to dogs and being outside are very strongly protected against a lot of these diseases. And this is not a value judgment as to why being outside is better for you. It's just a reflection of the fact that we have evolved by being outside and being present in these systems. And now through our time of evolution, we have not adapted to this changed lifestyle. Conversely, again, we're thinking about timescales. Our microbiota has adapted really rapidly because it only takes them in the orders of tens of minutes to replicate. So our microbiota is adapted to this changed lifestyle, but it's not producing compounds that are that healthy for us. So there's now this mismatch between our health and our biology and the microbiota and the lifestyle that we carry with us. Here we wanted to understand where S24/7 fit in this picture. And in the literature and through our studies, it became very obvious that industrialized populations have very low prevalence of S24/7. It's only about 10%. While it's very highly prevalent in other types of mammals, it's a very strongly tied, it's a bacterium or a family of bacteria that is very strongly tied to homeothermic animals. And we're very interested to find is that when we looked at traditional populations, S24/7 is present between 65 to 90% prevalence, indicating that it's a bacterium that it seems like we've been losing through our lifestyle. And while, again, we don't necessarily know whether it's good for our health, it's important that we think about some of the changes that will be accumulating due to this loss of organisms that is happening through our different lifestyles.
Carolina, one question. When you show, like with that previous plot, does this mean that everybody has that? I'm curious about this individual to individual variation. Does it mean that everybody has at 60%, or some people, or it's a fraction of people that don't have it? And same thing for the other species.
So we find that 60% of individuals have it at least at a couple of percentage of abundance. There's the reason why I said the level of the 60 to 90% prevalence is because we find reads for S24/7 when we look at the genomic sequencing in over 90% of the individual in the Hadza. While in humans that are industrialized, we find this only in 10% of the humans that we, the patients, or with the people that we've surveyed. So it's speaking of prevalence rather than abundance.
Got it. Thank you.
So here, it's as we wrap up, I think that one of the most important things that I think that this study has taught us, and at least has taught me, is this idea of how important the scales of these questions are, both in the terms of the time scales of, for example, how long of an exposure is necessary to lose some of these members, as well as the length scales that become important to really try to understand the system. It becomes really necessary to explore the system across the length scales. So here we talked about both S24/7 disappearance, and there's other members of the microbiota that disappeared. S24/7 was just the one that had the largest phenotype. We also find these changes in the mucus depletion that can have a lot of downstream effects. We find that, for example, after the PEG treatment, there's long-term targeting by the adaptive immune system to microbes that normally are considered to be commensal and good for health. We may be changing some of our interactions for the longer term. And also we talked about this external exposure being required for recolonization. And one of the things that I didn't have time to tell you about is that for this external exposure, it's very important that the habitat is back to normal. So if we do this recolonization of S24/7 when the system is still undergoing the osmotic perturbation, not surprisingly, S24/7 is not able to colonize. But importantly, these are the type of procedures that we do in the clinic tend to be at times of flare-ups. So when we think about fecal microbiota transplants, these usually happen at a time that the patients are having most of the inflammation. So we really shouldn't be that surprised that oftentimes these procedures do not work just because the habitat is not permissive for some of these species to actually be able to recolonize. And so to end, I'd like to thank the people that have been involved in this work. This is work that the lab just opened up about a year ago, and it's been a super exciting year with, of course, a pandemic happening in the middle, but I couldn't have asked to start with a better group, and we're really grateful for our funding. And thanks so much for everyone for listening, and I hope to be able to connect in person with all of you sometime soon.
All right, thank you so much.
That's great.
Yeah. So there are a few questions. One that is from Steph Weber is, in terms of differences in disease prevalence between traditional and industrialized populations, how do you separate effects from microbiota composition versus diet, exercise, etc.?
Hi, Steph. So that's a great question. Separating the effects is really hard. And in fact, separating the effects for all of these complex systems, we have to go to these mouse models in which we can start teasing things apart. One of the challenges with human studies is that even when we're looking studies for just diet, we can't have patients actually hold on to their specific regime just because it's hard, and we don't have the same constraints than, say, a mouse model will have. So some of the studies that are done to try to tease that apart are, for example, looking at instead of just the traditional populations, are populations that come from the traditional have a traditional lifestyle but live in cities, so that then you can start looking at there's only fewer parameters that are being changed. In those cases, one of the largest effects that we see from the microbiota tends to be changes in diet. There's some studies that are really great that look at how immigrants from different countries that are going to the US have a very changed microbiota, and within a couple of years of having moved, will have an impact on some of the diseases that they never experienced before. And again, here, there's a lot of things that are changed. There's not only going to be the changes in diet, there's also going to be the changes in lifestyle and all these things that get added together. And yeah, so basically the short answer is that we can't directly control this except for very short experiments or experiments with mouse models.
So I was going to ask a follow-up question that was suggested by Fred Cheng, and I was wondering the same thing actually, which is,
hi, Fred.
I have the strong impression that when we eat, just on the 6-hour time scale, that you're changing the osmolarity or osmolality of your gut. So that's what Fred was asking about. I definitely was wondering about the same thing. And yeah, and then this issue of time scale. Say it again.
Surprisingly not. So the change in osmolality will happen, for example, for people that are lactose intolerant. So in that case, then there is a strong change, and it comes with all the different GI symptoms, lactose intolerance. But for the most part, things are just very well balanced. Now, when we look at, for example, even bacteria that are growing in vitro, through the growth, we don't actually see large changes in osmolality. So there's still this balance that is being kept. So other things that can happen is if there's people that eat too much salt, will also have osmotic changes. But for the most part, most of the food that we eat will not have an osmotic impact.
There you go. So I got a question from Brandon Schloman, who's actually, he was a grad student in Raghu's Parthasarathi's lab at Oregon, looking at the microbiome in zebrafish.
Yeah.
And so what his question is, curious of your thoughts on details of the fairly rapid S2 24/7 drop. You measured reduced in vitro growth rate, but this is due to increased cell death or reduced division. Are cells dying rapidly in vivo due to PEG, or is their growth being stalled and then they are killed indirectly? Or are they being transported out of the gut and can't repopulate? So I can read that again.
So there's two things that happen during, well, there's many things that happen, but in terms of S24/7, we think that there's two main things that happen that lead to the deer's appearance. One of them is the fact that they're not growing. We think some of the cells are lysing. We're following up with some of these experiments in vitro, and we're depending on the strain, we're seeing that the cells are actually lysing, but not in large amounts. So we think that the main thing that that changes, and this we have to, we're just starting to do this on multiple strains. So some of the results are still on only a couple of the strains that I'm talking about. We think that it's mainly the reduction of growth. The motility of the gut is also increased because of the laxative being present. So we think that this effect also washes them out faster. And one of the things that we found is that even if we take some of the stool that has been passed during the time of high osmolality, and we use that to reseed the bacteria, the mice, they're actually able to take this in. So we think that there's at least enough of them being alive that they're able to repopulate the gut. Of course, we don't know how big the bottleneck is through inoculation and colonization, but there's definitely enough cells to be able to restart the system. The other thing that we're really curious about is whether the osmolality triggers some lysogenic events. So one of the things that happens, and I didn't have time to talk about this, but there's this very important relation between the phages that are present in the genome and how they're being triggered. So these lysogenic phages, we think that they might be triggered during the time of osmolality to actually lyse the cells and come out. And one of the things that we're currently investigating is trying to understand how other phages that are actually going through to the cells via these osmotic channels will be affected, because now you have a cell that is stressed and is producing more osmotic channels. And if through these osmotic channels we have the phages that are able to get in, you now get this other level of control that I think is really interesting, and we're just starting to delve into.
Great. One question, this is something I was curious about. Like in the earlier plots that you showed when you showed S24/7, so one question we're getting about it is, it seems that the total number of microbes present is constant, roughly the same. Why is that? I mean, I think there's the way I view it is some question about the carrying capacity of the gut, and right, who else is coming up when you stop detecting these guys and such.
So we definitely think that at least with low levels of these osmotic laxatives, you really are just changing the environment, and there are some bacteria that are taking advantage of it. This is not the case, for example, with the colonic lavages prior to doing colonoscopies. In those cases, you really are washing things out. But for mild diarrhea, it does not seem to be the case at all. You're just selecting for microbes that do just fine in this higher osmotic environments. And they're taking advantage of the extra food that is not being consumed by the ones that are having problems. So you really see these large blooms in E. coli, in the proteus, and a lot of the gamma proteobacteria. And one of the things that's interesting is that if we make the system less complex, for example, if we're just competing Bacteroideaceae and S24/7, so with one strain of the Bacteroideaceae are negatively impacted. So you do see that the load is reduced, but as soon as you add more members, you get to a constant level. There's a carrying capacity of the gut that you don't go beyond, and the members that are able to do well under stress just very rapidly bloom.
Cool. So another question, this is now regarding the mucus layer in the gut. Does an altered expression of mucin proteins affect the microbiota of the gut or water nutrient absorption?
So can you repeat that? The changes in mucus expression, you said?
Expression, exactly.
We looked at the level of, not directly at the level of mucus expression, but we looked at the level of mucus protein. And interestingly, that does not change. It just seems like it's being displaced from the epithelium. So it's just being scraped off by this increase in water flow. This is kind of the model that we have. When there are changes in mucus expression, the microbiota responds very rapidly. So there's changes in mucus expression, for example, because of the circadian rhythm, and the microbiota will respond to these changes. In fact, the microbiota sometimes has its own circadian rhythm matching that of the host. And in those cases, we don't really know what's the chicken and what's the egg. But in our case, we think that it's really the physical destruction of the mucus that is affecting the way that the microbes are able or unable to consume it.
So I'm going to have a go at it now. There's quite a few questions actually that writ large, I would say, have to do with the question of human health. Okay, and I'm going to give you the broad sweep, and you can pick and choose amongst them as you see fit. So there's questions about, are there psychological changes seen due to changes in gut microbiota? There's a question about opioids, which are known to cause constipation, and hence whether or not microbiota are affected by opioid. Ricky Garner was wondering about osmoprotectants that are genetically expressible. Then there was another one that had to do with metabolic syndrome being linked to diet and microbiome. Let me just see, I think I saw one or two more. Oh yeah, about probiotics, whether that's pseudoscience or something that should be listened to. So anyway, you can see, and probably experienced this lots of times, that people are quite curious in general about the health implications. So maybe you just want to riff on that a little bit.
Yeah, absolutely. So, the first disclaimer is that I'm not that kind of doctor. So whatever I'm telling you now, don't take it as a PhD advice as opposed to medical advice. So starting, I guess I don't remember all of them, but I'll try to kind of cover just in general what I think is important to think about. What worries me about the use of laxatives particularly is in pediatric populations. As I was mentioning, up to 2 to 3% of kids are exposed to these laxatives in a time that the microbiota is forming, and in a time that their immune system is becoming educated. And if you set up the community to be a community that is tolerant to these osmotic perturbations, you'll have unintended consequences. That you're selecting based on osmolality, but you also find bacteria that are good at responding to stress are not the ones that produce the compounds that we want to have. So what grandma said about eat your veggies very much applies here. And in the end, I think it's one of the best things that we can do to our microbiota is to eat lots of diverse types of fiber and eat lots of it. The standard dose that we are recommended is 30 grams. If we look at traditional populations, they eat upwards of 150 grams of fiber. So we're just in a very different regime. That said, if you have, for Western humans, if they eat more fiber, surprisingly, their diversity does not go up. We tend to think that increased diversity is it's very tightly correlated with better health. But unfortunately, because we're not being exposed to these microbes, we're not able to gain them back because they're not in our environment. So this gets us to the question of probiotics. The problem with probiotics is that they're the bacteria that we can grow, and they're not necessarily the bacteria that we want. So there was some studies that that were done by the LNF group, looking at how recovery from antibiotics was better or worse with probiotics. It seems like probiotics don't do that well, at least in the pill form. And I think that's because we're flooding our system with 1 billion of this bifidum, and this is a bifidum that was maybe isolated from a breastfeeding baby. So context matters a lot. And the bacteria that are good for me are not going to be good for everyone. So that's why it becomes there's a lot of historical effects, and that will change whether, if you take a probiotic, whether it's going to be able to embed, whether it's going to have an effect that is positive or negative on your microbiota. So unfortunately, this is not very satisfying, which is the reason why we're studying this at all, to try to get more predictability of these systems. So in general, I think that probiotics are good for your immune system when they're taken in a natural context. So it seems like eating yogurt, fermented food, those type of things seem to have a positive indication with some immune parameters. And again, like, these are all the simple things that everyone has always considered to be tied with health, right? Eating fresh foods, eating foods that are not over processed. And here we're starting to understand the mechanisms as to why that is true. Then there was a question on metabolic syndrome.
Yeah, let's see. Actually, we moved it over to the answer, but hold on. Let me see if I can find it. Metabolic syndrome has been linked to diet and microbiome and to heritable traits. How can we reconcile this question of genes versus lifestyle when it comes to microbiota? I think that's super interesting.
Yeah, that is super interesting. So there's other diseases that have a mild genetic component. For example, inflammatory bowel diseases. What I think that is happening there is that there's a predisposition that gets exacerbated by specific microbes. That there's, depending on which microbes you have, they may metabolize the drugs that are taken differently. So someone had mentioned opioids. Of course, those cause constipation, but anything we take by mouth will affect our microbiota. And in our microbiota, because whatever it produces ends up in the bloodstream the same way that a drug that you take by mouth would, it's affecting cognition in the same way that is affecting gut health. So I do think that it's something that, I don't think it's something that one should stress a lot about, but I think it's something people should be cognizant about. There's now starting to be a lot of ties with, also with someone I think had mentioned pain. There's a lot of nerves that end up in the gut. Some of the bacteria can signal, for example, if there's specific foods, there's this very interesting relation between not exactly what we think. I don't think that our microbes necessarily change the way we think, but they change the type of inflammatory molecules that might end up in the brain. So right now, the field is very hot, which I think is actually does it a disfavor because there's a lot of hype around it. I don't think that this is going to be the panacea that will solve everything. But I think that it's exciting because it's something that we can actually modulate. So I think that before we can use this for personalized medicine type of approaches, there's a lot more that we need to learn. But there's no way that these systems wouldn't be affecting all the processes in our body just because there's not really a filter for what they produce and what ends up in the blood system. So
yeah, okay, I think that's a good place to wrap up. It's great to see you, and thanks again for a really fun talk. It's cool to see where you've taken this, and as I said at the beginning, I really like the way you've sort of linked your work with KC and your postdoc stuff, and maybe even your undergrad days. So that'll be great to see how this continues to evolve. So I just wanted to let everyone that's left know that tomorrow morning we have Hopi Hoekstra from Harvard, and she, as I've already mentioned, gives fantastic talks. I don't even know what she's going to talk about, but she's one of those people, whatever she's going to talk about, it's worth hearing. So Saturday, I hope you'll be able to make it. And Carolina, thanks again. It was really great to see you. And
thanks everyone. And feel free to email me if you have any other questions or follow-up comments. I'd be glad to hear from you all.
Yeah, that's great. That's really generous. Okay. Well, see you guys tomorrow. Thanks again.
Bye, everybody. Thanks, Carolina, again.
Ciao.